Start कर दो।
जी. Assalamualaikum, I'm Dr. Sana Amjad, and I'm here to discuss a very important module of MRCOG Part 1, and that is genetics. So genetics is actually a very important module in your exam, and they usually ask about almost 8 to 10 questions from this module, and these questions have actually more weightage in your exam than even the anatomy questions. So you have to have attempt these questions very correctly, and they are very basics. You just need to have a very basic, you just need to have understand the basics of genetics. So we are going to start our today's topic, and this is genetics. And in this discussion, we will discuss about the chromosomes, and then we will discuss about cell cycle. You all must have read it that previously, the cell cycle, which is mitosis and meiosis, and then we will discuss about aneuploidies and aneuploidy syndromes, and then we will discuss the screening tests, diagnostic tests, and then last we'll discuss about the modes of inheritance, and that is very, very important in this genetic topic. Okay, so starting first topic, and that is chromosomes. So you all have known the chromosomes, and you all have seen the chromosomes. So chromosomes are actually the linear strands of DNA that contain genes, regulatory elements, and nucleotide sequence. So what is a chromosome? It has two structures. These are called chromatids, and the chromatids attach at the center, and it is called a centromere. Okay, so this upper is the short arm of chromosome. It is called P arm of chromosome, and there is a long arm of chromosome which is called Q. Okay, so each somatic cell in our body contains 23 pairs of chromosomes. So this is a very important MCQ. They can ask how many pairs of chromosomes in a somatic cell. So if they ask about pairs of chromosomes, then you have to tick 23. But if they ask you about how many number of chromosomes, then this will be 46 number of chromosomes. Okay, because 23 pairs means 46 number. Okay, so that's a very important MCQ that can also come. Now in 23 pairs, out of 23 pairs, 22 chromosomes are autosomal chromosomes, and the last pair of chromosome, it is called sex chromosomes. And you all have known that in female, the sex chromosomes are XX, and in male chromosomes, the sex chromosomes are XY. So coming to the size of chromosome, the largest chromosome is chromosome number 1, and going up till 22 chromosome, that will be the smallest chromosome. Okay, so that's very important that in our somatic cell, how many chromosomes do a somatic cell has? It is 46 chromosomes, like previously we discussed, 23 pairs and 46 chromosomes. And this is called diploid status. So somatic cells are diploid. Diploid means 2n, because every cell has 2 pairs of chromosomes. That is called diploid. Only the germ cells in our body are haploid, the ova and sperm, and they contains 23 chromosomes, not 23 pairs of chromosomes. This germ cells has only contains 23 number of chromosomes, because when sperm and ova, they fertilize, they form a zygote. So 23 chromosomes from sperm and 23 chromosomes from ova, they unite and form a zygote into a fetus that will be having 46 chromosomes, which will be normal. So germ cells have 23 chromosomes and they are haploid. Haploid means only one pair of chromosomes, 1n. So wherever this is written 1n, it means haploid. This written 2n, that will be diploid. So somatic cells are diploid and germ cells are haploid. Okay, so this is the basic structure of a chromosome. The chromosomes are divided according to the into different types of chromosomes according to their structure. Coming to this chromosome, it is called a metacentric. What does it mean? It means that centromere is at the center of chromosome, and both the arms are equal in length. Okay, so if the centromere is not at center, it's at close to one end, okay, one arm is short and other is long, then this is called submetacentric. And in third type, it is called acrocentric. Acro is very near, the centromere is very near to end, okay. It's a very short arm of chromosome, and that the second one is long arm. Only these three types, acrocentric, submetacentric, and metacentric, are present in human body. Okay, there are many more types which are not in human body. Like this is a telocentric. So they can ask about when the centromere is at the corner. Okay, there is no short arm. It's at a corner of chromosome. That is called telocentric. But this type of chromosome is not present in humans. Okay, you must have to understand this. So whenever they ask in humans, when the centromere is near the end of chromosome, which type is this? So this is an acrocentric type. So these three types are important to understand, and they can come in MCQs. And you can remember that how many acrocentric chromosomes are present in our body. There are actually 5 chromosomes and then last Y also. So 13, 14, 15, and then 21, 22 chromosomes. These 5 autosomal chromosomes and also a Y. So these 6 chromosomes, they only are acrocentric chromosomes. Right? Everything cleared up till now? So this is important chromosome structures. So coming out to our next topic, and which is a cell cycle. All of you must have read the cell cycle many times before, even in biology as well, in our metric and FSC. So we are going just to revise all our previous knowledge. So cell cycle is actually, what is cell cycle? It is a series of events in a cell. It leads to its division and replication. So you know the meanings of these: divide and replicate. So how the cell cycle works? It has 4 phases. Okay, that number 1, G1, it's a gap 1. Then S phase. S is called synthesis phase. And what is importance of this phase? It's a very important phase because replication occurs, replication of DNA occurs in this S phase. And then there is a G2 phase. This is also a gap phase. These 3 phases are combined and they are called interphase. Okay, you will just be seeing the word of interphase as well. So these 3 phases are called interphase, and then the last phase is mitosis. And you all know that cell divides in mitosis. So cell division is in mitosis, and cell replication is in interphase and S phase, during S phase. So this is a very important MCQ, and the MCQ comes very frequently, that in which phase of cell cycle the DNA replicates. So this is the S phase in which DNA replicate. Okay, and there are proliferation genes. Proliferation genes means that the genes which express to proliferate the number of cells. So cell division is increased. So these two are the proliferation genes, c-MYC and c-Jun. And the very important inhibiting gene is p53. So p53 is our inhibitory gene. It inhibits the division of cells. And you must have heard this p53 gene. This is a tumor suppressor gene. If it is not expressed, then the cancerous cells are produced because there are no check and balance of cell division and replication. So this inhibiting gene is very important. So this, coming to this figure, you see this is a G1 phase, it's a gap 1. Then this is an S phase, and then this is the G2 phase. So the cell enters first the G1 phase, then goes to S phase, and then goes to G2 phase. So these 3 phases are interphase. And you see there are multiple arrows which point the DNA damage checkpoints. in all the three phases. What they do, if the DNA is wrongly replicated, there is some mutation or there is some error in the DNA replication, the cell will not go into mitosis. Okay, the cell will go to apoptosis, so that the mismatched genes and the mismatched cells, they do not enter the division phase. So these checkpoints are very important for the cell growth. And you see, this is a G0 phase as well. It is called, what is G0? It is all called a quiescent phase. Quiescent means a resting phase. So if those cells which are not dividing, like you can see the nerve cells, they are not going to divide and they are not going to increase in number. So these cells, they enter into G0 phase and they are going to rest. Okay. So these two MCQs are very important. I have heard about these two MCQs coming on and off in the MRCOG exam, that they ask about in which phase the replication occurs, so S phase, and which phase is quiescent phase or resting phase. So that is the G0 phase. And when these three phases are done very correctly, DNA damage checkpoints, they have checked that everything is correct, then the cell enters into this M phase. And what is M phase? It is a mitosis phase. Okay, and in mitosis, the cell has to divide. Okay, up till now, everything is cleared. Now I am going to move to the mitosis. We will discuss a little bit in detail mitosis phases. So what is mitosis? Mitosis is actually a process of cell division that results in production of two identical daughter cells. One cell, it divides into two identical cells from a single parent cell. And what happens in this mitosis phase? This is the progenitor cell, 2N. 2N means the cell is diploid. Okay. Basically, every somatic cell in our body, they are diploid. So 2N is written. This cell, they enter the G1 phase. S phase means cell replicates its DNA. Then the G2 phase, these three phases are interphases, and then cell enter into the mitosis. So mitosis has these four phases: prophase, metaphase, anaphase, and telophase. PMAT, PMAT. Okay. So a diploid cell, it enters into mitosis and it generates two daughter cells which are identical. That's very important that these are identical. There is no change in the daughter cell, and the chromosome numbers are also same. Like the progenitor cell has 46 chromosomes, so is the daughter cell. Each daughter cell has 46 chromosomes. There is no reduction of chromosome number. Okay. So coming to the stages of mitosis, you just have to don't memorize everything. You just have to have a brief knowledge of this and what actually is being doing in these phases. Like these four phases: prophase, meta, ana, and telophase. So this is interphase. I have told you previously that interphase is all the three phases: G1, S, and G2. So during interphase, DNA is already copied. Okay. Now enters into mitosis. Then prophase phase, the chromosomes pair up. Look, the chromosomes, they are pairing up. And then the metaphase. During metaphase, nuclear membrane disintegrates. See, there is no nuclear membrane here. Nuclear membrane disintegrates and chromosomes line up at equator. They are line up here in the center. Okay, now in anaphase, what happens? These chromosomes, like this was the chromosome and the sister chromatids. These are sister chromatids. Okay, one sister chromatid and this is the second sister chromatid. They are pulled apart. So one chromatid moves to this pole and this chromatid moves to this pole. This is a very important phase and phase of mitosis, and we will discuss later on in the translocations as well. Okay, what happened in, no, not in translocation, in non-disjunction. Okay, so this is an important phase to remember, that in anaphase, the sister chromatids pulled apart. So what happened next? Then the telophase. In telophase, what happened? The cell actually pinches in the middle and it's about going to divide. Okay, and also cytokinesis, the cytoplasm also divides. And then the nuclear membrane reforms. See, nuclear membranes are reformed. So what happens is actually there are two identical daughter cells. Look, the progenitor cell, this is a progenitor cell which has 2n number of chromosomes. And look, the daughter cells, they are very similar, identical, and they have the same number of chromosomes. Okay, so the chromosome number remains the same. One cell divides and undergoes mitosis and give rise to two identical daughter cells. Okay, right? Okay, I'll move on to next slide, and that is meiosis. Now, what happens in meiosis? It is actually a type of cell division in which germ cells are produced. Only in our body, meiosis happens during germ cells production and division. Okay, so previously I have told you that only germ cells are haploid in number. There are no other cells that are haploid. So meiosis is also unique to germ cells. Okay, now what happens during meiosis? Look, this is a progenitor cell, 2n. It will be a stem cell, stem germ cell. It undergoes first phase meiosis because meiosis has two stages, 1 and 2. Mitosis has only one stage. Okay, so meiosis, there are two stages of meiosis 1 and 2. So first meiosis done, meiosis 1, during meiosis 1, duplication and recombination occurs. I'll tell you what is the recombination means, and it's very unique to meiosis as well. And then the meiosis 2 is separation of chromatids. And look, this was the one cell, which is 2n, and it leads to production of 1, 2, 3, and 4. 4 daughter cells, and which are haploid. So what happens during meiosis? The the chromosome number is reduced from diploid to haploid. So 4 haploid daughter cells are produced, and you must have seen that they are not identical. Mitosis, one cell give rise to 2 identical daughter cells. Okay, during meiosis they are not identical because there was a recombination. I'll tell you next slide what is recombination. Okay, so what happens in meiosis? In meiosis 1, basically chromosomes first, so सब में यही है कि chromosomes have to duplicate. Okay, then what happened is paternally and maternally inherited chromosomes are recombined. Recombined means like this is a paternal chromosome, one chromosome, and see this is a second chromosome. So what happened actually, that this one chromosome and this second chromosome, they cross over. You must have heard about chiasmata and crossing over. You don't have to go into detail, but this crossing over and this recombination between the non-homologous chromosomes, one is paternally inherited and one is maternally inherited chromosome, they cross over some genetic material to this. So this crossing over means that recombination has occurred. Some genetic material has recombined, so the daughter cells will not be identical. Okay, so diploid number is reduced to haploid number, and in meiosis 2 there's splitting of chromatids. So 4 daughter cells are generated from one dividing germ cell. Okay, paternal and maternal genetic material is mixed. This is called mixing. Okay, this recombination is called mixing, and each germ cell then has only 23 chromosomes. Like, see this. You don't have to memorize this whole chart. I'll tell you what is going during meiosis. We have understood that during interphase, the DNA is copied. See, this is a progenitor cell. It is 2n. And these, all the 4 phases like mitosis. So meiosis 1 and mitosis all has same phases: prophase, metaphase, anaphase, telophase, PMAT. So in meiosis 1, what happens? Prophase chromosomes pair up. See, this is the pairing up. That chromosomes here, they are recombined. Okay, so this is unique to meiosis. This does not happen in mitosis. So recombination occurs in prophase 1 of meiosis. Then the same is metaphase, that they line up at equator. The same is anaphase, that chromosomes are pulled apart to the mitotic spindle. Same is the telophase, they're going to separate. And there are two daughter cells are produced, which are haploid. Here, the daughter cells are haploid. Chromosome number are reduced. Now again, meiosis 2, the same occurs. Same occurs, the prophase, then metaphase. Metaphase line up at equator, then the anaphase, and then the telophase. So see, every cell, these are the 4 daughter cell are produced, granddaughter cells we can say. Every cell has single copy of chromosome, so every cell are haploid. And if you see, just see the chromosome, every chromosome has a different genetic material. If you just see the color of chromosome, this is a dark blue, this is a light blue, and these two has mixing of chromosomes. So all the daughter cells, they are not identical because of this recombination. Okay, so this is all about mitosis and meiosis. You don't need to memorize each and every phase, but you just have to know what happens during meiosis and what happens during mitosis. This is important, that recombination only happens in meiosis. There is no recombination in mitosis, and 4 daughter cells, they are not identical, and every daughter cell has 23 chromosomes. Okay, and that will be the germ cells. So germ cell are haploid, and they will be having 23 chromosomes. So in meiosis, the diploid number reduced to haploid number, but in mitosis, the chromosome number remains same. Diploid number divides, and the same 2 identical diploid daughter cells are produced. Okay, so these are just 2-3 differences between mitosis and meiosis. You just have to have a good understanding of these. Okay, so coming on to a very important topic, it's aneuploidy. So what is aneuploidy? Ploidy means chromosome number, and aneuploidy is an abnormality. So any abnormality of chromosome number is known as aneuploidy. And why there is abnormality of chromosome number? What is the mechanism of this abnormality? So what happens is, the very common mechanism of aneuploidy is non-disjunction. Now what does it mean? It means that pairs of homologous chromosomes at meiosis 1 or sister chromatids during mitosis, anaphase. Now this you have to know, okay, what is being doing in anaphase. So the homologous chromosomes or sister chromatids, they do not separate. So both chromosomes are passed to one daughter cell and none to other. So I will go back to the anaphase. See, this is the anaphase. Okay, what happened during anaphase? That chromosomes pulled apart. So these are 2 chromosomes, right? And each chromosome has to go to one mitotic spindle and pass on to one cell. So each daughter cell has 1 chromosome. What happened in non-disjunction? They do not separate. They do not pull apart. So what happens? The 2 chromosomes, they do not separate, and they goes to one cell. One cell has a 2 copy of chromosome, and the next daughter cell, it's empty. There is no chromosome, this chromosome. So what happened? Like this, one daughter cell will have an additional chromosome number. So that chromosome do not have to pass through the daughter cell. So one will have an additional chromosome. It will called a trisomy. And the other one will have a missing 1 chromosome, and it will be called as monosomy. So basically what happened, that these 2 chromosomes, they have to separate and goes to one cell, one cell. Okay, if they do not separate and they just go to one cell has an extra copy of this chromosome. This should not be present here. This X should not be present here. This is an extra copy of chromosome. So non-disjunction means non-separation. You can just easily remember that the chromosomes do not separate, and they pass on to one daughter cell, and this daughter cell is empty of that chromosome. So what happened is, this is deprived of one chromosome number. One chromosome is missing, one minus, you can say, and this cell has one extra copy, so 1 plus. When it will be 1 plus, then it will be called, because there are 2 chromosome numbers, so trisomy, and which one is absent, then that will be called a monosomy. So this is a basic mechanism of trisomy conditions. You all must have heard very important trisomy conditions. These are very important in Down syndrome. You all must have heard about the Down syndrome. That what is Down syndrome? It is actually the trisomy of 21 chromosome. Now, what I have told you earlier, that this trisomy means that only in 21 chromosome number this trisomy occurs. That 21, the babies with Down syndrome, they have one extra copy of only 21 chromosome, not the whole. Like, for example, sorry. Like what happened in the karyotype. So we have a normal karyotype of 46XX. It's a normal female karyotype. So what happened in Down syndrome? Down syndrome, they have just a extra copy of 21 chromosome, not the whole lineage of chromosomes. Only 21 chromosome is there's an extra copy. So what happened is 46, then plus 1. Okay, so Down syndrome will have a karyotype of 47. If it's a female, it will be XX, or male, it will be XY. So there is an extra copy of chromosome here. Then there is Edward. Edward will be having trisomy of 18. Patau will be having trisomy 13. So Edward, you have to remember these very easily. Edward start with E, and 18 start with E also. So Edward syndrome trisomy is 18, and Patau syndrome trisomy 13. Then there is a Klinefelter syndrome. It is 47XXY. Now here 1X is extra. So there is non-disjunction of X chromosome. So 1X, there is an extra copy of this X chromosome. So karyotype is 47, and this is a XY. This is the karyotype of Klinefelter syndrome. It's a very important karyotype. They can ask about Klinefelter syndrome karyotype. They can ask about Edward syndrome karyotype. Like if they ask what is the karyotype of male Edward, so male means XY, because there is no abnormality in sex chromosomes. So XY to male rahega. And what happened in Edward, there will be a 47 chromosome. Okay, so in monosomy, one copy is missing. So in Turner syndrome, it's very important that 1X is missing. So it is X0, only 1X. And the number is reduced, not 46, they are 45. Okay, so you know now that what are the trisomies and what are the monosomies. The trisomy, there's extra copy of one chromosome, it will lead to 46 to 47, and then the monosomy 1 minus, so 45 karyotype. Okay, so move on to the second mechanism of aneuploidies. First mechanism was non-disjunction, which was non-separation. Then there is another mechanism, it is called translocation. It are 2 types of translocations. You just have to have basic understand these things. You don't need to memorize these translocations. I will tell you what to be asked in the exam. So reciprocal translocation is actually the chromosome rearrangement involving the transfer of genetic material between 2 non-homologous chromosomes. So that's just the transfer of genetic material between 2 non-homologous chromosomes. This is called reciprocal translocation. I'll tell you here in this picture what is the translocation. See, this is a normal arrangement. this picture, this chromosome, this partner, it's a normal arrangement. And what is this? What happens in this parent? Like this is the normal, this is normal, and what happens between these two chromatids of non-homologous chromosomes? These two are non-homologous chromosomes. They are not similar. Okay. So what happens there is the genetic material is exchanged between these two chromosomes. This dark brown has to be here and this light yellow has to be here. So this is the reciprocal translocation of genetic material, right? Okay. So the most common reciprocal translocation in humans, this is important. You have to remember this, that most common reciprocal translocation, not this number, but only this, that it is between 11 and 22 chromosomes. So the translocation of genetic material from 11 to 22 chromosome, it occurs. This is a very common reciprocal translocation in humans. Okay. Now, what are the two categories of this translocation? There is a balanced translocation and there is an unbalanced translocation. I'll tell you in next figure. Balanced means it does not have phenotypic consequences because the adult or child has a normal amount of genetic material. The amount of genetic material does not change. So they do not have any phenotypic abnormalities. But in unbalanced, there will be one chromosome with missing genetic material, like monosomy, and another chromosome with duplicated genetic material, like trisomies. So what happened, like in non-disjunction, this also in the unbalanced translocation. See this figure again, this diagram. This parent was normal arrangement, and this is the chromosomes that have a reciprocal translocation with a balanced translocation. What does it mean? Balanced means if you see the normal arrangement, these two materials, the genetic material remains the same. There is no extra copy here. So this is the balanced. And what happens when these two rearrangements in their children, these two partners they mate, and then the children, what happens is what are the possible outcomes of the offsprings? They can have a normal chromosome rearrangement like one parent. It's normal, totally normal. Okay. And what happens here? This and this. You see, this was one parent and it's also balanced translocation. Or the chromosome number is same. There is no extra genetic material. And these two are unbalanced. You have seen, you see how these are unbalanced. See, this is an extra. This yellow, this much genetic material is extra here. These chromosomes are normal and this yellow is extra. And what happens here? This was missing. This yellow has to be have here. So this offspring, they have a missing genetic material. So these two are unbalanced. Okay. So pregnancy can have four possible outcomes. I told you in the figure. One possibility is that a normal pregnancy with a normal karyotype, like this. Then the normal pregnancy with a familial balanced reciprocal translocation, like this. The balanced translocation, it does not matter. This is normal pregnancy will occur. Now, what happens if these two are unbalanced? Unbalanced have two possible outcomes. Either they can go into the spontaneous miscarriage, or if they goes to term and the baby is alive, so the child will have a high likelihood of learning difficulties and congenital anomalies. Okay. So these are the four possible outcomes in the translocations. So all the trisomies and the monosomies, they basically have the same mechanisms of aneuploidies. So one was the non-disjunction, then second was the reciprocal translocation. Now this is the second type of translocation. It is the Robertsonian translocation. Now what happens in the Robertsonian translocation? It is a chromosome rearrangement and it involves fusion of long arms of two acrocentric chromosomes. You remember we studied the acrocentric chromosomes. There were 5 acrocentric chromosomes. Also, the Y chromosome is also acrocentric, but in autosomal pairs, these 5 chromosomes are acrocentric: 13, 14, 15, 21, and 22. So what happens in Robertsonian translocation? I'll tell you in the figure. See this diagram? These are, they have shown the acrocentric chromosomes. Like centromere is near the near the end, and short arms, they are not visible actually here, but only the long arms are visible. So what happened in acrocentric chromosomes? This long arm of one chromosome, it moves to this non-homologous chromosome. So this brown chromosome, it moves to this yellow chromosome. So this is called, when the two acrocentric chromosomes, their long arms fuse. That is called the Robertsonian translocation. Okay. So what was studying in Robertsonian only involved the 5 acrocentric chromosomes, because only the acrocentric chromosomes, they have the long arms of chromosomes. Now you have to remember this, that the most common Robertsonian translocations are, not remember this 13q, 14q, but this you have to remember that fusion of chromosome 13 to 14 and 14 to 21. Now you remember what was the trisomy of 21 known? That was Down syndrome. So 14 to chromosome 21. Whenever 21 chromosome is involved, it will lead to Down syndrome. And this 13 chromosome, this Robertsonian translocation, it will lead to Patau syndrome. So 13 and 14, they are fused, and 14 to 21. So these translocations, non-disjunctions, they are the basic mechanisms of all the trisomies: the Down syndrome, Edward, and Patau. Okay. So this also has 4 possible outcomes. Like previously, we understand of reciprocal translocations here as well. If there's a normal parent with normal arrangement, and this Robertsonian translocation. Now again, this will be one balanced, or this is unbalanced. Balanced means the number of chromosome or the genetic material remains same. Just this arm of chromosome, they move to this arm. Okay, so there is no extra add or subtraction of chromosome. So this is a balanced translocation. So balanced translocation in one parent, and this parent is normal chromosome. So what happens? What are the possible outcomes in offspring? So first outcome, the normal pregnancy with a normal karyotype. Like this parent, this is a normal rearrangement. Then there's a normal pregnancy with a balanced translocation. So this is a balanced translocation, like one parent, balanced translocation. And then they are unbalanced. Now see how it is unbalanced. See here, this yellow, this chromosome is extra, okay? And here, this brown chromosome is extra. So these are the trisomies. So what happened in unbalanced translocation? Either this spontaneous miscarriage attributable to an unbalanced translocation, or spontaneous miscarriage or to an imprinting defect, or if it go unbalanced translocation, it goes to term, a live-born child with multiple congenital abnormalities will occur. Okay, we will discuss about the congenital abnormalities of these aneuploidies later on. So you just have to have a basic understanding of these translocations and this non-disjunction. We just need to remember what is the most common translocation, like this 13 to 14, 14 to 21 in Robertsonian, and in the reciprocal translocation 11, 22. Okay. So moving on to our next, these are the aneuploidies you have to have remember. Sorry. So what is the Down syndrome, Edward, Patau, and Klinefelter? So one by one we will discuss. We all have discussed Down syndrome. In Down syndrome, there will be trisomy 21. Only the 21 chromosome will be extra. No other chromosome will be extra, okay? It is prevalent, it is 1 in 700, and causes. See, most common cause is non-disjunction. You have now know What is non-disjunction? It is 95% cause, and of the maternal gametes as well. And then there's a Robertsonian translocation, 3%, and then there's a Meiosis as well, 1%. So outcome, because Down syndrome is actually compatible with life. So babies are born with some learning difficulties. There's a moderate learning difficulties, not very severe. Now you will get a question like they will give you the facial features of syndrome, and they will ask you what could be the possible syndrome of this baby. So Down syndrome has very typical facial features. You have to remember these features: upslanting palpebral fissures, epicanthic folds, flat midface, and brachycephaly. Means AP diameter is smaller. The head is smaller in the AP diameter. These are the facial features that are very important. And duodenal atresia, it is also an Hirsch sprung as well. These are also pathognomonic of Down syndrome. And atrioventricular septal defects, they are very common in Down syndrome as well. Okay. So coming to the Edward syndrome, Edward is actually the trisomy of 18 chromosome number. It is less prevalent than Down syndrome, 3,000, and the causes are same in all three trisomies. There's a non-disjunction, Robertsonian translocation, non-disjunction translocation. In Klinefelter, only non-disjunction occurs. Okay. So Edward syndrome, they actually do not have a good outcome. If the baby is born with Edward syndrome, otherwise there are more chances that they die uterine. But if they deliver, if they are born alive, then very chances that they will be die within first year, even within months. Very commonly, most die within months. So they are not have a good outcome of Edward syndrome. And the facial features of Edward as well: micrognathia, cleft lip, cleft palate. Now you have to remember there are few pathognomonic. So the clue words, no? What is here? What is in the Down syndrome? Down syndrome, these palpebral fissures, epicanthic folds, even duodenal atresia is also very pathognomonic of Down syndrome. But what is Edward? These 2 points, they will give you the rocker bottom feet, clenched hands. These two are very important. If any MCQ has this rocker bottom feet, you have to go for Edward syndrome. Okay, most of the things, they are same in even Edward and Patau as well. So cardiac defects, they will be cardiac defects very commonly, and there will be exomphagus also, GIT abnormalities, muscular abnormalities, facial abnormalities. There's so many abnormalities in these trisomies. Okay, so moving on to Patau syndrome, it is actually 13. It is actually a 13 trisomy, 1 in 5,000, same causes, or isme most likely they end up in spontaneous pregnancy loss. Very, very few they born alive, and even then they die before their first birthday. Okay, so in Patau syndrome, what happened? This is important. Midline defects. They will give you midline defects. And holoprosencephaly. It is a very important. Holoprosencephaly, what does it mean? We'll discuss in embryology as well. But prosencephaly, holoprosencephaly is actually that the prosencephalon, जो हमारा forebrain है, it will not develop into the two hemispheres. So the midline defects are common in Patau syndrome. There's a holoprosencephaly, then there will be cleft lip, cleft palate, cardiac defects, polydactyly. It's also very important for Patau syndrome. So these few clue words you have to see in the MCQs, and then you have to go for it, either Edward syndrome or Patau syndrome. Okay, and lastly is the Klinefelter syndrome, and we have discussed previously that Klinefelter, there is extra copy of X chromosome. Okay, and this is also very common, 1 in 1,000 prevalence, are almost similar to Down syndrome. Non-disjunction is the most common cause, and outcome, they are not diagnosed prenatally unless you are doing some invasive testing for some other reason. But you cannot diagnose it prenatally. The baby boy, these will be the male. So male babies, they were born very normal, and you don't know that they are Klinefelter syndrome unless they are grown up and you do their infertility workup. They will be infertile. There's azoospermia, they'll be infertile. You go for their hormonal workup, and then you see that they are the hypergonadotrophic hypogonadism, and then you will go for karyotype, and you will see that it's a Klinefelter syndrome. So these are not diagnosed earlier unless you go for their karyotyping, and that will be done during infertility workup. So in this, you see there's a tall stature. You can get MCQ on this as well. There's a tall stature, the infertility workup is done, and only there's a slightly decreased IQ. Okay, so these 4 syndromes, they are very, very important to understand, and you can get one MCQ from these 4 syndromes as well. Okay, so coming to their screening and testing of these aneuploidies. This is actually a risk assessment. You can say a risk assessment of Down syndrome, of chromosome non-disjunction. Like in age 20, you have a risk of 1 in 1500 is a very less risk of chromosome non-disjunction, means chromosome abnormality risk. So what happens here, like in after age 36? See, 36 age, you have a risk of 1 in 300. And so the cutoff for invasive testing is 1 in 250. Basically, this cutoff actually, it changes for invasive testing because currently we are having some more non-invasive testing as well. But this is considered as high risk if the risk is 1 in 250. So 36, 38, almost in during this, between this, the 37, after 37 age, these are the high risk for chromosome abnormalities. So age is a very, very important, significant factor in chromosomal abnormalities. You see, advanced maternal age is a very important risk factor for chromosome abnormalities. If the age of mother is 45, then the risk of having the chromosomal abnormalities is 1 in 30. It's a very, very high risk. Okay, so you have to know just these few points in this table, that what is the risk in 40 age, 1 in 100, you can remember, then 45, 1 in 30. Okay, and you can remember 36 or 35, you can say 1 in 300. So these 3 age, you have to remember these. Okay, so first is the risk assessment due to age. Okay, then moving on to other screening tests, what we do for the screening of these aneuploidies. Okay, so screening actually starts from the first trimester. You all have known what is the first trimester up till 13 week of pregnancy. So what we do during first trimester, we do a dating scan, and we do scan for knuckle translucency as well. So what is knuckle translucency? It is a sonographic appearance of subcutaneous fluid at the back of fetal neck, okay, between weeks 11 to 14 weeks. So this time duration during first trimester is very important. So first trimester dating scan is very important. You have to do knuckle translucency during this scan. So NT, if it is more than 3.5 mm, then it will be high risk. Okay, you can have the aneuploidies in the baby. So what do we do next? In the first trimester, we do 2 things. First is the knuckle translucency ultrasound. The second is we do biochemical screening. Biochemical screening means we take blood sample and we send these biochemical markers. So you have to remember this test, combined test. Combined test is actually the 3 testing. One is NT. NT is knuckle translucency. Then beta HCG, and this is PAP A, pregnancy associated plasma protein A. So you have to remember like this, NBP. Okay. Okay, what is combined test? Combined test is actually has three components: NBP, N is knuckle translucency, beta SG, PAP A. So you have to remember what happens in Down syndrome. Like in Down syndrome, babies, knuckle translucency is increased NT. Okay, then beta HCG, you send biochemical marker, it will be raised, and only this PAP A is reduced. Okay, so these are very important biochemical markers for screening. These are just screening tests. You cannot diagnose that, oh, this is Down syndrome in baby. No, these are screening tests. You screen that, oh, this, if these are raised, NT is raised, beta CG is raised, PAP A is reduced, then you can say that, oh, this pregnancy is a high risk of developing aneuploidies. You cannot say it is a Down syndrome or it is an Edward syndrome. There are few changes between the syndromes that only in Down syndrome beta CG raises. Otherwise, Edward and Patau, beta CG reduces. So you can say by seeing these biochemical markers as well, but still this is a high risk for developing aneuploidies. You have to confirm these by doing the diagnostic test. We will discuss later on. Okay, so first trimester, you have to do these two tests. NT combined test means combined test is also includes NT. Okay, so don't consider this extra. And during second trimester, you have to do these two tests. So first trimester is 13 weeks. Up till 14 week, you can do combined test. And from 14 week onward, you can do triple and quadruple test. Now, one is triple test. Triple test means three biochemical markers, A, B, E. You have to remember these by heart. And quadruple means fourth one is added. So A, B, E, and I. If you see these all are in the alphabetically order, that alpha fetoprotein A, B, E, and I. You can change that order. It's not mandatory. This just to memorize that this triple test is A, B, and E is added. Estriol, quadruple test में inhibin is added. Okay, so I'll move on to the next slide. Now, this is a very important table in your essentials book. You have to remember what you have to remember in this table. See, in first trimester, you doing an NT scan. If it is raised, effect in trisomy 21 pregnancy, only Down syndrome pregnancy is considered here. If it is raised, so detection rate is 70 to 75%. Okay, and there are few ultrasound markers of Down syndrome as well. We are doing ultrasound, we are doing NT scan. Okay, ultrasound, we do NT scan on ultrasound. Then on ultrasound, you do NT plus nasal bone measurement. If nasal bone is absent or hypoplastic, it is also very, very associated with Down syndrome. So detection rate is increased. 80 to 85% chances that it will be trisomy 21. And if the third thing is added, that assessment of blood flow in ductus venosus. So if it is absent or reversed A wave in ductus venosus, then also the detection rate is 80 to 85%. So ultrasound also can diagnose 80 to 85% chances of trisomy. Okay, then this is a combined test. You see, this is N, B, and P, and T is raised, beta G is raised, only PAP A is reduced. This combined test, this figure you have to remember by heart. That combined test has detection rate of 90%. So this is a very important thing to understand. Okay. Then the next is a triple screen. I have told you alpha fetoprotein, beta CG, estriol, quadruple test, alpha fetoprotein, beta CG, estriol, inhibin. So if you see in Down syndrome, what happens is actually beta CG raised and inhibin is raised. These two things are raised. PAP is reduced, estriol is reduced, alpha fetoprotein reduced or unchanged as well. Okay, so triple screen, they have 70% detection rate, quadruple 80%. So what has the most detection rate? It is the first trimester combined screening. Okay, so don't need to remember this. It is very easy. The triple is 70, it's a 70%, and 80% and 90% is combined test. Okay, now there are a few tests, not more, but it's additional, that it is integrated test. What is integrated? Integrated actually means you combine two tests. One is combined test and then quadruple test. You just add two things that you are doing in first trimester biochemical screening as well in the second trimester. Okay, you do both testing in the patient. So you do anti-PAP A in the combined testing, and then you do quadruple screening also 86. And the sequential means that first you do combined test. If it is raised, then only you will do quadruple. So this is a sequential testing, that first you are doing a testing in first trimester, which is combined testing, and then you are doing a quadruple testing as well to increase the detection rate. See, detection rate increases to 95%. What was the detection rate of combined test? It was 90%. And if you add after combined test and then you also add the quadruple test, then the detection rate is increased to 95%. Okay, so these two, I think, detection rates are important, and you have to remember what increases and what decreases in Down syndrome. One thing is very easy, that in Edward, everything reduce. You don't need to remember anything. Everything reduced. Not anti. Anti is already anti will be raised. All reduced biochemical markers. Okay, only biochemical markers like inhibin reduced, estriol reduced, beta-HCG will be reduced. So beta-HCG is actually a differentiating factor between Down and Edward or tau. So Edward में भी beta-HCG reduce होगा, but tau में भी beta-HCG reduce होगा. Only in Down syndrome it will be increased. So these are few differences between these syndromes. They can ask you, otherwise they ask you about the Down syndrome, and it is very important that only beta-HCG and inhibin is raised. Okay, baki estriol and alpha-fetoprotein PAP A, they are all reduced. Okay, so move on to our next topic, and it is a very, very important topic. What's this? It is a non-invasive prenatal testing. The word shows that it is a non-invasive. We do not have an invasive testing, and the prenatal testing, NIPT, we call it NIPT, and it's also called a cell-free CFFF. DNA means cell-free fetal DNA. Okay. Now, what's the basic mechanism of this cell-free fetal DNA? Basically, what happens in the mother plasma? Cell death occurs, and cell death results in the release of DNA fragments from nucleus to the plasma. So maternal plasma have different DNA fragments in her plasma. So what happened is the cell-free DNA in maternal plasma is a mixture of DNA fragments arising from dying cells in the mother and placenta, because when the woman is pregnant, the dying cells of mother as well as placenta, because placenta is working as an apoptotic organ. So DNA fragments of mother as well as fetal DNA, they will be present in maternal plasma. So the proportion of fetal to total cell-free DNA, it is called as fetal fraction, is actually about 10%. Matlab in maternal plasma, the cell-free fetal DNA is actually 10% is of fetal fraction. The fetal DNA fragments they are. So we can detect many things from that fetal DNA by just taking maternal plasma. Okay, so free fetal DNA is released by apoptotic cells in placenta. So its composition is therefore representative of the nuclear content of cells in the developing fetus. So whatever apoptosis is occurring in the fetus, it will be released from the placenta into the maternal serum, and you can detect this fetal fraction by just taking maternal plasma. Okay. So what is the significance of this prenatal testing? See, in pregnancies with fetal trisomies, okay? Trisomy means one extra copy of chromosome, like down, Edward, down. What is happening that the number of fragments derived from extra fetal chromosome. See, जो extra fetal chromosome होगा, जैसे कि like 21 chromosome or 13 or 18. So the number of fragments derived from this chromosome will be higher than in diploid pregnancies. So the fragments of that DNA, which was an extra copy, that will be more. So we can detect from the maternal plasma. So previously, currently means that cell-free fetal DNA, it can be tested to determine the gender. Like you can get the karyotype as well. It is XX or it is an XY. And then the rhesus blood typing, like RH status of the fetus as well. Now recently it is being using for the detection of trisomy 13, 18, and 21. So this is a very, very important non-invasive prenatal testing. And see its detection rate, it's 99%. So it is a very, very good screening method. And you can have first the those screening tests like combined test or triple or quadruple. And if you have a testing of that it is a high risk of developing the aneuploidies, then you can go for cell-free fetal DNA. Okay, but again, this is just a screening test. This is not a diagnostic. Okay, you have to remember this. Whenever they ask you about diagnostic test, then this is not a cell-free fetal DNA. It is not a diagnostic test. It is screening test. So this screening testing, its advantage is that its detection rate is increased 99%. So if the previously your combined testing or triple testing, they can be false positive in that as well. So you can go after, if that testing was high risk, you can go to this testing, NIPT testing. If this remains high risk, then you have to go for diagnostic testing as well. Okay, and many times you can they cell-free fetal DNA, they say that it is normal, so you don't need to go to the invasive testing. So this is a very important and very brief topic of cell-free fetal DNA. And coming to the diagnostic tests, so you all have read it many times, that what are the diagnostic tests of all the aneuploidies, all the abnormalities of chromosomes. See, these three are very important. This is the chorionic villus sampling, amniocentesis, chordocentesis. So what happens in chorionic villus sampling? It is CVS. You have listened multiple times. So in CVS, you aspirate placental tissue. So chorionic villi. You take a sample of chorionic villi, it is a placental tissue. You can take it through the transabdominally, or you can take it transvaginally, ultrasound guided. Okay. So you take a sample of placental tissue, and you take two testing. In the CVS, you take two samples. One is you take cytotrophoblast in placenta, okay, and also you take the mesenchyme as well. So cytotrophoblast are very actively dividing cells, and you can have a rapid result. Because they're actively dividing, you can have a rapid result in 2 to 3 days. And in mesenchyme, you have to go for culture. So the best advantage of CVS, there are two advantages. First is the rapid testing, okay, rapid result in 2 to 3 days. And if this is positive, you have to wait for culture as well, because culture will be the definitive diagnosis, okay. And second is the time duration. It is being doing in first trimester, 11 to 14 weeks. You cannot perform it before 11 as well. If you perform it before 11, then the fetus will have limb abnormalities as well. Okay, so this is the best time, 11 to 14 weeks. This is a very early time for testing of any abnormality. So these two are very advantages of this CVS. And miscarriage rate is actually same in all three, 1%, and it is dependent on the trainer. So the operator dependent, their miscarriage rate. So that there's no difference in all the three testing. So what is the next? Next is amniocentesis. The name shows that you aspirate amniotic fluid. So amniotic fluid you aspirate, and you culture amniocytes within it. Amniocytes are released from fetal skin, urinary tract, GIT. So you take amniocytes and you culture them. So culture result again, it will become in 1 to 3 weeks. So it is time taking, and you can get this testing after 15 weeks. You cannot perform it before 14 weeks. Okay. So whenever the scenario comes with the scenario came, the pregnancy is high risk and the pregnancy is 16 week. Okay. 16 week up till now, and then you get her biochemical screening, combined testing or triple testing, they came the high risk. So what will you do next? Okay. How will you diagnose? Means if they ask you about diagnostic test, then only you have to select these three options. Not chordosynthesis actually we consider for these abnormalities, only CVS and amniocentesis, and you select the CVS or amniocentesis by seeing the gestational age. If it is 16 week, means if it is after 15 week, you have to select amniocentesis. Okay. If it is between 11 to 14 weeks, you have to select CVS. Even in 10 week, you can select CVS. Okay. No, but not before 10 week as well. So this is the amniocentesis, same as carriage rate. Now, what is it? The chordosynthesis, the name indicates that it is the sample of the cord, umbilical cord. So fetal blood is taken from umbilical vein, and it has to be happened after 18 week. It is actually very difficult to do a chordosynthesis. So training has to be procedure. Training is mandatory for this procedure as well. So it's not very common, and you have to take the culture, and result will take 1 to 3 weeks. So mostly the question comes for chorionic venous sampling and amniocentesis. Chordosynthesis we actually do when we have to have detection of fetal anemia. So if you want to see the HB of fetus, like in the intrauterine transfusion as well, so then only the chordosynthesis can be done. Okay. Then that time you cannot do the CVS or amniocentesis. So chordosynthesis, the main purpose is in detecting fetal anemia. Okay. So clear up till now the diagnostic tests of all these aneuploidies. First step is the screening test, then you move on to the diagnostic test. You cannot jump straight to the diagnostics test. It's a step-by-step procedure. In the first trimester and the second trimester, if the abnormality is diagnosed in first trimester, like combined testing result is high risk, then you must have to go to diagnostic test in the same trimester. Like you can do a very early CVS can be done in 11 to 14 weeks. Okay. I hope everything is cleared up till now. So moving on to our next very, very important topic. you must have two or three questions must come from this topic, modes of inheritance. Like what is the mode of inheritance of this disease? What is the inheritance pattern of this disease? So there are few modes of inheritance. To autosomal. Autosomal means the disease involves the autosomal chromosomes. The 22 pairs are autosomal chromosomes. So disease pattern is actually a dominant or a recessive pattern. Dominant means the capital. You can write like the capital. Recessive, it's a small d. Okay. Then this is the X-linked disease. If the disease involves the X chromosome, then it has two patterns again, X-linked dominant, X-linked recessive. And then the third inheritance pattern is mitochondrial inheritance. It's a very less common, and we don't have get any question from this. But you have to know what is mitochondrial inheritance. You must know, okay, mitochondria, we get mitochondria only from our mother. This is the basic concept of our biochemistry as well. So you get mitochondria from your mother. So every disease which is mitochondrial based, it will pass from mother only to all children, whether it is son or daughter. Okay. So mitochondrial inheritance means the disease passing from mother to, if the disease is to the father, disease present in father, it will not go in any of the children. Okay, this has to be understand. Okay, so we will discuss all these pattern one by one. I will discuss with you. Okay, so what is autosomal dominant? Autosomal dominant means it caused by disruption of only one gene of a gene pair. Okay, there are two genes of every chromosome, so only one gene is disrupted. Okay, I will move on to next slide. Actually, I don't go for to memorize these pedigrees. Okay, I will tell you how you can memorize, how you can solve these pedigrees. So if autosomal dominant, I'll tell you in detail. This is a normal gene. Like this is a normal gene pattern, so small d. Autosomal disease dominant means that if one gene is dominant, I will write it's a capital, so you can say this is a dominant gene. Okay, so if one gene is present, then the disease will present. So this gene pattern, it will be the disease or affected, and this is normal. Okay, so these two patterns are in the autosomal dominant. See what have they discussed here in this affected parent and unaffected parent. So this is unaffected, normal, and this is an affected parent. Okay, so what you have to do to solve a pedigree, you cannot memorize each and everything. You just take an affected parent. If they give you a scenario like the one parent is affected and other is normal parent, then how the disease pass on into the children. So see, you just have to combine these two testing. Like I will tell you in again. See, the affected parent, this is affected parent, and then this is unaffected parent. Okay, so you just have to combine all these genes. This capital D is a dominant gene, diseased gene. It combines with this small gene. Okay, just write. Don't think anything. Then again, this capital D, it combines with this D. It again make this pattern. Then turn off this small d. Okay, this small d combine with this small d, this and this small d, again this small d. So what you have seen here, we have discussed that this small are normal. Okay, these are absolutely normal. And what happens here, this capital D, these are diseased, affected. So what happened? There are two possibilities. If one parent is affected and other parent is normal, what are the possible outcome? What is the possible outcome? See, 50% chances that the children will be diseased and 50% chances, sorry. Now I have to draw it again. What's happening with this autosomal dominant? Oh, okay, it's written. So what happens? It's actually the 50% are diseased, means 50% are affected, and 50% offsprings are normal. See? So coming back to our previous, or you can see here as well. What's happening with this? Okay, so this is the autosomal dominant pattern. I'll tell you on this figure. If one parent is affected, other is unaffected, then what are the possible outcomes? 50% chance the child will inherit the gene, mutated gene. 50% chance the child will not inherit the gene. So 50%, 50-50 हो गया, ठीक है? So, हाँ ये यहाँ पे आना था मैंने. So what happened is autosomal dominant. Only one gene disruption. So males and females are equally affected. Why? Because this is a disease of autosomal chromosome. So there is no discrimination between male or female. Only the total percentage. Inheritance is 1 in 2. 1 in 2 means 50%. We have drawn a pedigree, and there you have seen that 50% chances of having a disease. And that's important. Individuals in each generation are affected. Even if one parent, whenever there is one parent affected of autosomal dominant disease, the disease will go into 50% children. Means chances are that disease will go on to the next generation. If the disease goes into next generation, then there are chances that disease will go into the next generation as well. So there is no skip generations. Every generation, they have individuals with affected disease. Okay, it's written like this in pedigree. See, this the filled one are affected. It's affected male, affected female. They just have tell you, but there is no discrimination between male and female. Yeah, they're just telling you that say this generation is affected, then the next this is also affected. Then again these are affected, then again this is affected. So every generation has got a disease. Okay, so this is an autosomal dominant pattern. Now what happens in autosomal recessive pattern? Autosomal recessive. Now this is caused by disruption of both genes in a gene pair. Okay, both genes. Recessive. Ab recessive ko I'll write with a small letter. Now see, this is a diseased individual with autosomal recessive. So it is a diseased. Now there are two possibilities in autosomal recessive. What is the possibility? If the two mutated genes are present in an individual, then the patient will be diseased of autosomal recessive. But if there is a one normal gene and the second recessive gene is present, this will be a carrier. Okay, a carrier status is only present in this autosomal recessive pattern. Okay, so what's normal here? You can write, say this is normal here. This is the normal gene, then this is the diseased because this is a recessive pattern. This I'm writing just for an example. Okay, there is no authentic thing that this is capital is always normal. Okay, it's just I'm writing to explain to you that this capital means that it is normal for autosomal recessive. Okay, recessive means a small genes, no? So recessive means the two genes have to be present to express the gene. Okay, so if the one gene is present, then this will not be evident in phenotype, and the disease will be patient will be carrier. Again, males, females affected equally. Now inheritance pattern. You cannot remember this inheritance pattern for autosomal recessive. You can only remember inheritance pattern for autosomal dominant because there are two possibilities. Either the patient is diseased or the patient is carrier. Then the possibility is different. Okay, I'll move on to the next pedigree for autosomal recessive. These are few examples I'll tell you to memorize. There's nothing to. Okay, so you cannot memorize these pedigrees. You just have to have a basic knowledge of these recessive things. Okay, what happens is here. I told you there are three possibilities. This person is normal. Okay, if there is a one recessive gene, it is a carrier. See, if the both genes are recessive, then this will be diseased. Okay. Now you have to write these three things first, then solve the question. Now, see if question they give you that the both parents are carrier. Question they give you the both parents are carrier. You just take out this genotype here. One parent, father or mother, both, and other parent. Okay. So both parents are carrier. Now you have to draw a pedigree, like I tell you previously. First take this capital D and mate with this T, then take this T and this. Okay, what happened in this? Capital D, and capital D with small d. You don't need to think anything here. You just draw this pedigree. Okay, so you have taken this gene. Now take this gene, the small. Now take small gene and compatible with this capital D, small d. I will write like this, capital first. Okay, just to take it easy. Then the small d with small d. Okay. Now look at this corner and categorize these three things. What is this? See, this capital D means that this is a normal. So it is a normal. And what is this capital D, small d means? This is a carrier status like the parents. So these are the carrier. And what happens here? This is important. They actually ask you this, that what are the chances that if both parents are carrier of this disease, autosomal recessive, they don't give you that this disease is autosomal recessive. They just give you the example, like cystic fibrosis. If both parents are carrier of sickle cell or any cystic fibrosis, like sickle cell, the both parents are carrier with sickle cell disease. Okay, what are the chances that their offsprings will be diseased? So this small d, this is the diseased. So what chances? If you take 1 in 4, 1 in 4 means 1 in 4 chances that the offspring will have disease. 1 in 4 means 25%. So 25% chances that the offspring will have disease, 25% chances it will be normal, and 50%. There will be 25%, so combined they make 50%. So 50% chances that the offsprings will be carrier. Okay, so they can ask you anything from this pedigree. You cannot memorize each and everything here. You just have to draw this pedigree. Okay, you just have to have a basic understanding. They can ask you, how if the both parents are carrier, how many chances that the offsprings are carrier? So now you just draw it and you see 50% chance that the offsprings will be carrier. And how many chances of normal offspring? Normal 25, and disease will 25%. Okay, so I will remove everything now. Everything, I hope कि clear हो गई है यहाँ तक. This pedigree, this they are telling you in this that see, this is an unaffected. Affected is only this. Okay, all the rest generations, they are unaffected, but these dots show carrier. See, one generation is carrier, has a carrier status. Pass the carrier gene to the next generation, again to the next generation, and then the disease appear. So when the disease appears when two carriers they meet. One carrier cannot pass the disease to the offspring. Okay, so if you excuse me, Okay, one thing again, I'll tell you, if they give you a scenario that one parent is carrier, carrier and other parent is normal.
Blue or azul? Blue azul. Blue azul.
Sorry for interruption, students. Just give me 5 minutes. Just give me 2 minutes. Okay. I'll So I was giving you another scenario for someone recessive. Like what happens if they give you that one parent is carrier, okay, and the next parent is normal. See, one is carrier and the other is normal. So what are the genes of carrier? Carrier means they have one small gene, and normal means there is no small gene. So what happened in this scenario, that the disease will not be apparent. Okay, so to just write this capital D, then this again this capital D, and the small d, capital D, capital small. So what happened here, that 50% chances that the patient will be normal. So not patient, the offspring will be normal, and the 50% chances that the offsprings will be carrier. So there is no disease. So if one parent is carrier, the disease will not pass. There will be no disease in the offspring. Only 50% are carrier and 50% are normal. Okay, so this pattern actually happens when so this is called the skip generations, that if the disease is evident one after many generations, you can say after many generations. Okay, so that was the table. We are coming back here. So autosomal recessive, disruption of both genes, equal affected males, females, inheritance 1 in 4 when the both parents must be carrier. So the both parents will be carrier, then only the disease will be apparent in the offspring. 1 in 4 means 25%, not when a single parent is affected or carrier. So only one generation is affected. There will be the skip generations as well. So what happened in X-linked recessive? Now, X-linked recessive, it means the disease is pattern on X chromosome. So recessive mutation is present within a gene on X chromosome. So what happened? This is the X chromosome, and the female has both 2 X chromosomes, male has 1 X chromosome. So if the gene, this recessive gene is present in male, like this small d, recessive is small, so only one gene is present and the males will be affected. Okay? And for the female to be affected, there have to be 2 genes. Otherwise, if only there is one small gene, then the females are not affected. Okay? So only male individuals are affected, no male to male transmission of condition. Because what happens is actually, see, this is the diseased male, and the partner is normal. You see, you can see that this is normal, capital D, and this is small d. Okay, so if the disease is present in the father, like male, then what happens in his offsprings, that only in male, the male fetuses will get Y chromosome from father, not X chromosome from father. So that means that no male to male transmission of condition. Okay, so male offspring will get Y chromosome from father and X chromosome from mother. So that means that all the male fetuses will be normal. So in one generation, males are affected, next generation, males are normal. Okay. So this small d, this will go to the females. Okay. And the disease will not be evident because this is recessive. They have to be present two genes, two recessive genes have to be present on females to be expressed. So condition may seem to skip generations as well, because the disease, one disease in father, then the disease will not be in the sons, also not in the females, not in the daughters as well. Okay, the daughters will be carrier, only carrier. The carrier daughters will again give the disease to grandson of this male. Okay, if I'll write it again, this is very haphazard, I think. See, I write it again. So what is X-linked recessive? You just have to have a brief understanding. This is an XX and XY. I'm telling you that this is a diseased father, okay? And mother is normal. I'm taking example of hemophilia, okay? Hemophilia is X-linked recessive disease, okay? And hemophilia, mostly males are affected because the females have to have two genes from two disease genes, then only the disease expressed in females. So what happens here, males, this is the father, this is the father, and this is the mother. Okay, so make a pedigree of XX. This is a, you can say, a carrier female. This X again to that X, this also a carrier female. Okay, and this Y will combine with both of these X chromosomes, so all the males will be normal. 2 times I will write. See, what happens is that these are daughters, these are males. All the males means 100% males are normal. Okay, 50% of offsprings you can say, but this is X-linked disease, so you have to specify about son and daughter. So 100% males, sons are normal when the father is affected with hemophilia disease. But the females will be carrier. Okay, so this carrier status, this will not be apparent in this generation. So disease is here, but disease is not in this generation. Again, this carrier female will pass on this gene to his son because she has this small h, and whenever this small h, it will combine with Y chromosome, so the son, his son, will have the disease. So it means skip generation. One generation has the disease, daughter does not have the disease, grandson will have the disease. Okay, so this is the pattern of X-linked recessive disease and skip generations. In the X-linked, coming to the X-linked dominant disease. Okay, X-linked dominant means X disease will be SA dominant. Even if the one gene is present, the disease will be dominant, and like this will be the male. But they are saying that this dominant gene, it manifests so severely in male fetuses that they result in spontaneous loss of pregnancy or early neonatal death. So X-linked dominant disease will not be evident in male because they turn into spontaneous pregnancy loss. Okay, it only be evident, so it seems to be restricted to female individuals only. And again, the dominant disease, this is a pattern of capital D, so all the dominant diseases have an inheritance pattern of 50%, 1:2, but only of affected females. Okay. So coming on to the last inheritance pattern, the mitochondrial, I told you previously, they're only transmitted through the maternal line and can affect both sexes. Okay, so this was the basic concept of all the diseases. You have to draw the pedigrees, you have to understand these pedigrees because you cannot memorize these things. That इतने percent chances हैं कि diseased होंगे इतने carrier, so you cannot memorize all these things. You have to memorize these things because you must know what disease are autosomal dominant, what disease are autosomal recessive. This I just put the table from your book. You don't need to go into detail of these symptoms, these clinical features. First, you have to know what disease condition, what's inheritance pattern of these conditions. So tuberculosis, Marfan syndrome. Marfan syndrome is important. It's come in exam as well. And breast cancer. And it's very important. BRCA1 and BRCA2. Mostly they ask about what is the inheritance pattern of BRCA1 and BRCA2, and another important is this percentage. Lifetime risk of having breast cancer with BRCA positive, and lifetime risk of ovarian cancer with BRCA positive. So you can say roughly it is 20 to 40, and it's doubled for breast cancer, 40% for ovarian cancer. Okay. Then this is HNPCCC for the colon cancer, endometrial, gastric, ovarian. Also, all the cancers, these two actually are cancers. So cancerous conditions are autosomal dominant. Then the Huntington disease, autosomal dominant polycystic kidney. Now, it is also very important. Polycystic kidney has two subtypes. This is an adult. Adult one is autosomal dominant, but infantile, there's also an infantile polycystic kidney. That's autosomal recessive. Okay, you just have to memorize these things. Chondroplasia is also autosomal dominant. Then I have told you this autosomal recessive inheritance pattern with the pedigree. And then again, these conditions are autosomal recessive. You have to remember the cystic fibrosis, muscular atrophy, TOACS disease, hemochromatosis, trypsin deficiency, congenital adrenal hyperplasia, and deafness. I have jotted down the most important you have to memorize. I'll tell you in after a few slides. Okay, these are X-linked recessive disease. This is important. DMD, Duchenne muscular dystrophy. This is hemophilia A. It's important. I gave you this example. Okay. Okay, then albinism, X-linked dystrophy, fragile X syndrome. It is important. What's inheritance pattern of this X-linked recessive condition? And then there are a few X-linked dominant conditions: incontinentia, pigmented Rett syndrome, and vitamin D rickets as well. 3 conditions. This I have jotted down all the most common examples of all the disease. So I have highlighted the ones which are very important. In autosomal dominant, you have to remember chondroplasia, Marfan syndrome, polycystic kidney. Which one is adult type? BRCA1 and 2. Okay. Then autosomal recessive. These 3 are very common: sickle cell, cystic fibrosis, thalassemias. Okay. Then X-linked recessive, hemophilia and DMDs. They are very important, and neurogenic diabetes insipidus as well. X-linked dominant, they can ask you about vitamin D resistant rickets. Mitochondrial inheritance is not actually these disease important. Just have to memorize these names of the diseases. Okay, they can ask anything else, but you have to memorize these important examples of these syndromes. So this was all about today's lecture. And coming to the few questions, you have to memorize these, or if there's any question, you can get it. Everything is cleared up till now. Do you have any question? Or I have written down just a few questions. You just have to see these examples, like they ask you about what type of inheritance pattern does red-green color blindness follow. So you all have know up till now that what type of this color blindness is this. So this is an X-linked recessive. Okay. And again, the sickle cell disease pattern, you have know that this is an autosomal recessive, but they ask you about single gene or polygene. So single gene. So sickle cell disease is actually single gene autosomal recessive disease. And then they ask you, that was the question I was telling you before, that age 40 years, this is a high risk, 13 weeks pregnant, found to be high risk for Down syndrome following the combined screening test. See, it has undergone the combined screening test. Most appropriate further tests to see if the fetus is affected. See, you have to diagnose that fetus is affected or not. So at 13 weeks, which diagnostic test you go? You go with the CVS. Okay. So this was the basic concept of okay, this is a very common question. If both parents have beta thalassemia minor, now you have to know that what is the inheritance pattern of beta thalassemia. This is an autosomal recessive. Okay. If both parents have beta thalassemia minor, minor means carrier status. Both parents are carrier, then what is the chance of their female offspring having beta thalassemia minor? So they have asked you about carrier status. So we have done it earlier that if one is carrier and the second parent is also carrier, so what are the chances? This one is normal, this one is carrier, again this one is carrier, and the last, this one is diseased. So 25% normal, 25% diseased, and how many carriers? 50%. So that's how you draw a pedigree. You don't need to memorize anything from these pedigrees. You just have to memorize that what is the inheritance pattern. This is important. Which disease has which inheritance pattern? That's the only thing you have to memorize. So they ask you about offspring having beta thalassemia minor, means carrier status. They are asking you 50%. If they ask you how many chances of having a diseased baby, diseased means beta thalassemia major. If they ask you about beta offspring chances of having beta thalassemia major, so major will be having 25%. Okay. So coming to that's it for today's lecture. If you have any questions, you can ask me. Any question up till now? No question?
बस फिर ठीक है डॉक्टर सना, इसको ना हम वाइंड अप करते हैं, ठीक है?
Okay, ठीक है. Okay, thank you. Okay, thank you everyone.